We want to welcome you all. I'm Ruth Lehmann, and I'm one of 3 co-organizers. Together with Shoshi Yoshida and Erez Ras, we have been putting together a schedule, and this all started actually at a lovely meeting, and now it seems so odd. We were all sitting at a table together, sharing a meal in Münster. To sort of work out the program, and that's over a year and a half ago now. And we're really excited because, and I think that's a little traditionary for this meeting, basically everyone said, yes, we want to come. And so we really hope we have a fantastic program that every one of you is now, unfortunately not here in person, but listening, will find something that is new, that's exciting and relevant for them. And so we usually start off with a keynote, and I'm actually amazed that Allan hasn't given a keynote at this meeting yet, or maybe he has, I don't know. No, so because, you know, if there is one person who is all about the germline, it's Allan Spradling. So I want to give you a little bit background of Allan. So he grew up in Michigan in Kalamazoo, and he actually studied mathematics and physics at the University of Chicago. So then he changed from physics, going to MIT as a graduate student with Sheldon Penman, and really got into the early days of molecular biology, but already applying it to more developmental or organismal context, looking at heat shock genes. He then, and this starts the love for the ovary, as a postdoc, he moved to Indiana to join Tony Mahawal's lab. And this started an analysis initially focusing on one of the subjects that he has made enormous contributions, and that is on, we could call it genomic integrity, we could call it genome regulation and genome function, and that was mostly about replication and gene amplification using the chorion genes, and then also the other genomic regions. What many of you also associate Allan with is his incredibly fruitful collaboration with Jerry Rubin, which led to the P element, using the P elements as a tool to introduce DNA into the Drosophila genome, and then also to use that for single gene mutagenesis. And that clearly, and I have benefited enormously from that, established Drosophila beyond being a great developmental genetics organism, but really as a premier organism for doing genetic analysis of development. Allan also contributed to the Genome Project. In addition to the work on genome amplification, Allan really, when he started to work on germline stem cells very early on, when even we knew very little about what stem cells really are, he really established the ovary as a model to study the the fundamental principles of stem cell biology. What are aspects of self-renewal, differentiation, de-differentiation, what's the niche, what's the influence of nutritional growth? All of those questions were splendidly addressed and defined by Allan. And more recently, he has been looking more at oogenesis and how the metabolic state and challenges and aging affect the conditions of oogenesis. He also has made contributions to mouse germ cell biology. And just like for the fly, he just addresses the critical questions and does the critical experiments. Often we call, you know, like the killer experiments, which really solve certain problems. In mouse biology, his work has really clarified the role of the cyst development, follicle cell biology for the ovary, and in particular, talking about defining experiments, he really showed the lack of adult stem cells in the mammalian ovary. Allan has been an HHMI investigator since 1988. He has he has always had a relatively small group, which is typical for being a Carnegie investigator, small but exquisite. He has trained an enormous number of researchers who have gone all over to many universities, and many of them are possibly on this call or on this Zoom. Allan has been awarded many different prizes. He received the Conklin Award from Society of Developmental Biology, the Genetic Society Award, the Beadle Award from the Genetic Society, the Gruber Prize in Science, and most recently in 2018, the March of Dimes Prize in Developmental Biology. I want to just conclude by saying one thing that's my favorite quote from Allan, where he told me once, you know, everything that's touched by the ovary just gets better. And with that, I think we're going to hear more about oogenesis now.
Well, thank you very much, Ruth, for that very warm and wonderful introduction. I'm just putting my screen up, and I hope you can all see it. I'm really honored to be giving the opening talk in what I consider to be, you know, the best meeting that I tend to ever go to. It's just always so interesting. And what I want to tell you today is, since the time is short, is just to give you some general thoughts, share with you some general thoughts on germ cells, and also highlight that with some recent work from our lab. So just to mention, germ cells arose with animals, and all animals have them. They seem to be an innovation that's necessary when the genome size increased substantially to you know, hold the extra information needed for metazoan development. Let's see if I can.
Excuse me, Allan. Can
Yes.
you go to full screen? Thank you.
How's that?
Just fine. Thank you.
Okay, so, oops. So how do germ cells do this function? Well, one of the indications of the universality of germ cells is the fact that there's a core set of genes that are shared by germ cells in all the animals. Some of these are shown here. They're genes that are very familiar to everyone at this meeting. Some of these genes, for example, set up the piRNA system, which is very important for controlling transposable elements. But another universal and perhaps less talked about aspect of germ cells is this program of generating interconnected germ cell cysts by synchronous division and incomplete cytokinesis. Here you see cysts forming that are going to give rise in the testis to cysts that stay throughout testis development. It's very likely that this force behind the evolution and maintenance of the cyst program is the suppression of meiotic drive, another really serious threat to germ cells, that because these elements are constantly evolving. The system seems that the protection seems to work by having sperm develop from mRNAs that are transcribed at the diploid phase, and then a translational regulation is used, including by some of the conserved germline genes, which also form RNP particles and regulate mRNA activity. If there's any additional later after meiosis, if there is haploid gene activity, then the cysts come into play by sharing those products among all the developing sperm, so that it's very much harder for one genotype to get an advantage over another genotype, because whatever they try to make becomes shared with their potential target. Interestingly, this cyst paradigm comes into play in some gonads that we're very familiar with. Here's some figures from a paper in 2009 that showed that the earthworm gonad forms by cyst divisions in which as shown in the upper right, the cytokinesis furrow splits the pre-existing ring canal. And this gives rise to a gonad with a central core and cells all around it that open into the core And this same system is used to generate the familiar C. elegans hermaphrodite gonad. Well, in females, cysts have facilitated another development. So in many species, instead of just making oocytes, they have another cell type that comes from the cyst called nurse cells that probably, here you see some images taken from papers from a whole wide phylogenetic range of ovaries as illustrated in the bottom, all of which have nurse cells and oocytes. What these functions of the nurse cells are in all these diverse systems still awaits study. However, a brief history of what nurse cells do in different groups and how it's changed in evolution is illustrated on this slide. In the upper portion, you see a diagram of the zebrafish oogenesis, which is a good indicator of kind of the ancestral role of nurse cells, where they form just prior to meiotic entry. They stay around during the early parts of meiosis, but then they've turned over by the time the follicles have formed, and much of the growth occurs after the nurse cells are not present. About 250 million years ago, at the start of the Mesozoic era, the four clades of higher insects came up with a really impressive innovation to this system. Namely, instead of turning the nurse cells over early, like in the traditional system, they're maintained throughout oogenesis and produce almost the entire oocyte. And this greatly speeded up the ability to make eggs. And obviously these groups are among the most successful. But an even greater modification that has been the one that took place in about 160 million years ago, giving rise to the placental mammals that we're very familiar with, such as the mouse. And here, actually, the production of yolk, the nurse cells are still doing their traditional early job. Then there's a reduction of the yolk production stages, and a great deal at the end of oogenesis and the first few days of embryonic development has to do with making the nutritive coupling to the mother. And this caused the onset of germ cell development and embryonic development to be delayed by 5 or 6 days after fertilization. So a very talented postdoc in my lab, Steve DeLuca, wanted to understand more what kind of cells are nurse cells really. They traditionally had been thought of as germ cells, but in many ways they're acting like a somatic cell in Drosophila in helping to synthesize much of the oocyte contents. So to test this, he both developed a system using silencing reporters that I won't have much time to talk about, and also showed that you can isolate pure germline stem cell nuclei, the cells that precede the cyst divisions, and also each stage of nurse cell development, and look at their chromatin. The reasoning is, if we look at the polycomb silencing by looking at polycomb domains shown here in these green bars for this particular region of chromosome 3 that's illustrated, then you should be able to tell if a cell is a differentiated cell by whether the polycomb domains have become silenced and shown high levels of H3, K27, Me3. When you look at the starting chromatin in the germ stem cell, you see a very uniform distribution of this modification. And you can contrast that with these 2C or 16C follicle cells where the polycomb domains do have high levels of modification. And these are actually summarized here by this binning system below. And then when you look at late nurse cells, you see they look just like the differentiated cell, like the follicle cell. And in data that I don't have time to show you, this actually arises very early in the development of the follicle. So the nurse cells differentiate and acquire both physical silencing and polycomb modification during the very earliest stages of follicle development. Now this probably is similar events are happening in the Drosophila embryo diagrammed here. Data from Lee et al. Shows that prior to cycle 14, there's also this kind of broad chromatin modification pattern, sometimes called non-canonical chromatin, and you only get polycomb after cycle 14 in high levels in the polycomb domains. And the same thing appears to take place in the mouse, where the oocyte and the early embryonic stages have a broad distribution of polycomb, and later, which here is just illustrated by an ESC line, you get the higher levels of modification here in the HOX-A complex and also in these individual polycomb regulated genes. So one of the questions we're very interested in is, what is the function of this non-canonical chromatin in the germline cell cycle? But a question that this system is very good at addressing compared to the embryo is what actually initiates the onset of differentiation and of polycomb silencing in particular. It's difficult in an embryo with both maternal and zygotic gene regulation to do all the gene manipulations you'd like, and the embryo is so complex and there's multiple cell types, it's hard to get the earliest stages of any one cell type. All that is greatly simplified in the germline, where you just have the germline stem cell and the major product is the nurse cell, and you can isolate all the intermediates, and the genetics is simple. And by going through a lot of the polycomb related genes, which are diagrammed up here in A, the PRC1, the PRC2, and the various proteins, and here's some gene expression data. And I'm not showing you, but if in Steve's paper that came out last month, you can follow this in much more detail, and he's also available for further discussion throughout this meeting on the Slack channel. But he found that among all these genes, there's two that change in a very dramatic way, illustrated here in C. So these are the tips of ovarioles, and the stem cell and very earliest regions are kind of shown in this pink bars. And then as you get into the nurse cells differentiating and repression beginning, you have the purple bars. So what you see is that this gene polycomb-like, which is an alternate subunit of PRC2, is very high in the stem cells, and then it goes down during differentiation. Conversely, the SCM gene, which is this bridging protein that can bring either PRC2 or PRC1 to pre's and to the domains, is almost off in the earliest stages, but then comes up during differentiation. And so really it's these changes we think are what regulate the onset of polycomb repression. The PCL actually probably gives a lot of the character to the non-canonical chromatin state. It's thought to slow the dissociation events so that a few sites get highly modified, but because it has a very broad DNA binding specificity and the time is slow, there are probably actually different sites in different cells. And as a result, or maybe at least our observation is that these GSCs show a bivalent pattern of histone modification. But once SCM is there, their PRC2 is taken to the, focused on the domains, and then they're fully modified. Well, this is, this process of differentiation is essential for oogenesis. And you can see that here in the middle, where here's a low mag just view of two Drosophila ovaries, and then here in Hanser of Zest, the mutant, the catalytic core of PRC2 has been removed by a germline specific RNAi. And what you see is the ovary is tiny, it's sterile, and if you were to dissect that and look, you'd see that the follicles are only developing up to the stages where polycomb differentiation begins. And molecular analysis shows that PRC2 is both providing a general repression in the black and the green chromatin. And it also has a few specific targets outside of these regions, some of which are important possibly for controlling genes prior to sex determination, such as chinmo, and some other targets. So it's interesting. It's the role seems to be fairly limited and broad, but it's crucial. And another related function is H3K27 acetylation, which is carried out by this NEDG gene. And if that's mutated, you get follicles forming, and then they just can't grow. And that makes sense because the nurse cell program involves a very high upregulation of growth, metabolism, MYC dependent, and many of these genes that are upregulated have to be acetylated, and that's blocked without K27 acetylation. Well, I want to turn now, how similar are these nurse cells and this function that we've characterized to mouse nurse cells? What I'm showing here is just a kind of diagram showing our current limited, but this is what, by studying it, we see happening in the mouse ovary between the time the germ cells arrive, the PGCs arrive at the ovary at E10.5 and the completion of follicles at P5. You get a fragmentation of initially 32 cell cysts into multiple smaller cysts, and then there's a kind of slow process where the cells get smaller. They seem to transfer cytoplasm and organelles to neighboring cells, and eventually you end up with about 6 oocytes. And this same process happens in the two types of follicles, both the medullary follicles that develop immediately, called wave 1, and the cortical follicles, which develop more slowly, make the pool of follicles that are used throughout life in a mammal, called wave 2 follicles. Well, to try to investigate this, another really outstanding postdoc in the lab, Wanbo New, carried out an extensive single-cell RNA-seq analysis involving 52,000 cells throughout the period of time from when the germ cells arrive at the gonad to when the primordial follicles form. Here you see just the germ cell plotted, the pattern of sequencing the germ cell stages that you see, and not too surprisingly, you go 0, 1, 2, 3, 4, 5, 6. That's just the premeiotic and then the major well-known stages of meiosis at the right times that previous studies had shown that these are the predominant stages. But there's also this cluster 7. And cluster 7, unlike these others, comes from a variety of times. These others are all in a very narrow time window. But you see this magenta part of the bar graph is cluster 7, and these are the nurse cells, or the cells, the dying nurse cells, but they die very slowly by a programmed cell death, not classical apoptosis. And we think that this is because they're transferring their cytoplasm and organelles to the final oocytes, as shown here in this image, where the lineage labeled cyst fragment. This cell is going to be the oocyte, and then you see the green mitochondria and the orange Golgi elements moving into this, forming the Balbi antibody in the oocyte. And these cells, unlike in Drosophila, these cells can't be identified by RNA-seq immediately, but as they start to transfer and get smaller and transfer their contents, they show up as those type 7 cells. So what we see, what we've learned about those is that they have very low UMI because most of their cytoplasm is not present. And interestingly, they are arrested at a meiotic stage that's earlier than the companion oocyte. We noticed this from because we know the time when the individual samples were taken, and then the cluster 7 would have meiotic gene expression that was from the previous stage. And you can see that here in this image, where at this this is E19.5, but the oocytes are approaching the dictyate stage, which you can see here, but these nurse cells around it are still stuck in the diplotene stage. Well, we also were interested in the role of the somatic cells in the mouse ovary, as many others have been. It's much work, some of it which a few of the authors are credited below, have shown that the somatic cells arise from the coelomic epithelium. And there's two types of these cells. The early or bipotential cells, which arise even before sex determination, can differentiate either into Sertoli cells or into pregranulosa cells of this early type, which we're calling bipotential pregranulosa cells. And then somewhat later, although the exact timing of this has never been completely clear, you get LGR5 plus progenitors in the surface epithelium of the ovary dividing, and their daughters move into the ovary. And these labeled early BPGs give rise to wave 1 cells, but the EPGs labeled with LGR5 Cree will label only the second wave follicles. Well, to try to get some more resolution and insight into these pregranulosa cells, I'm showing you from the RNA-seq the pregranulosa cell and epithelial cell fraction of the cell analysis. And it suggests, interestingly, that these two cell groups that are going to make the wave 1 and wave 2, the pregranulosa cells, are really developing in tandem throughout much of the period of follicle development. Even by E12.5, there's clearly two groups, and each one of these two groups of somatic cells. Here's the dashed lines show that the time points 12.5, 14.5, 16.5, etc. And each time there's two, and each time there's one of them has the gene characteristics of the bipolar or bipotential, cells. The other more the epithelial, as you can see in some of these gene profiles here, where like FOX ligand 2 would be in the BPGs preferentially, and only in after birth do they express in all of the cells. And then LGR5 and GNG13 often label the opposite of the pairs. Furthermore, cell lineage experiments suggest that what you get in the way you get the distribution between the two follicles is that the BPGs initially associate with all the germ cells, but then the incoming EPGs from the surface displace them in the cortical region, but they don't enter the medullary region. Furthermore, although this differentiation is in tandem, and you can see that most of the genes expressed in these are the same, there's also some specific gene expression. And so we can get ideas as to how these carry out their functions in the two different lineages. And there's also interesting parallel here because there's two cell types, somatic cell types in the Drosophila ovary, the escort cells and the follicle stem cells. And the follicle stem cells replace the escort cells at about the point where the germ cells are in Pachytene. And that there's a general correspondence where the escort cells look like the BPGs and express some of the same genes, and then the epithelial cells look like the FSCs, look like the EPGs. So this potentially to us suggests that there's even greater parallels both in somatic and germ cells in the development of ovaries, and we're interested in learning more about that. And there's even potentially a second wave, first wave dichotomy in Drosophila, because it's well known that some of the germ cells never make it into the niches in the pupil or the late larval ovary, and they develop directly, and it's never been shown whether they really involve the follicle cells or if they develop just directly with the escort-like cells. Well, the last thing I'm going to talk about is just a surprising finding that Ethan Greenblatt made in the lab that suggests more similarities between the mouse and the fly, namely the great instability of the meiosis I spindle that's well known to be a factor in human aging of the ovary. And in the mouse, you also see a lot of aneuploidy in older mothers. It wasn't known that this would happen in the fruit fly, where eggs normally are laid immediately and they develop with a very high fidelity. but what Ethan developed a system where you could study aged ovaries and know how old they were, and he showed these curves of how fast the eggs go bad, like in 25 degrees, they go for about 2 weeks. What was only found out a little bit later is that what's going bad almost 100% is the meiosis 1 spindle, and that these are just curves of the arising of aneuploidy in these offspring of these embryos, suggesting again that this is the spindle, and this problem is perhaps much more widespread than just mammalian oogenesis, and is some kind of fundamental part of oogenesis. So I'm just going to summarize then to say that I've told you some generalities about early germline development. Nurse cells actually are somatic cells, and that there's a lot of similarities between nurse cell and somatic cell development in diverse groups. However, the nurse cells in the mouse seem to be quite a bit different, in that they probably never differentiate, because they're gone by the time that the Drosophila nurse cells would be differentiating. So there are also substantial differences that we need to understand better. And I'll just end there and take your questions. And thank you very much for listening to the talk.
Thank you, Allan. Please enter your questions in the question and answer. There's already a question from Chris Gonzalez, and she's asking, why do you say that nurse cells are somatic cells instead of just differentiated cells?
Well, to me, there's two kinds of cells. There's germ cells or somatic cells. And they don't look like germ cells anymore, but they have all the characteristics of somatic cells. And they just leave the germline later. That, you know, you can leave the germline lineage in the embryo, or you can go all the way almost to adult development and leave it at that point. So the point is that in terms of their chromatin and you know, kind of their strategy of gene regulation, they're following what seems to be a somatic program. And beyond that, you know, it's just a matter of terminology, so who cares?
So I wanted to know a little bit more about the phenotype of the EZs in the germline stem cells, because you said that they're, you know, they had sort of this very different chromatin state. I mean, it looks like they're almost not repressed because they have so little H3K27 marks. But when you took out EZ, you know, the ovary was very tiny, but what is the phenotype? So do the germline stem cells actually need repression at all, or is it really downstream of the germline stem cells?
Well,
the repression, which when the marks come up.
We were able to learn more about that than I had time to explain. But using our repression reporters, where we can put like a heat shock GFP gene all around the genome, and then when a region becomes repressed, like a polycomb domain or heterochromatin, you no longer can induce the gene. But in the germline stem cell, wherever you put that reporter, heterochromatin, rDNA locus, polycomb domain, they always are accessible and can be induced. And then you can see them turn off. First, the heterochromatin turns off prior to meiosis, and then during polycomb silencing, I told you the kinetics of the turnoff. So EZ doesn't seem to be required for the germline stem cell. It doesn't seem to be needed for the very earliest steps of going into meiosis, but it becomes essential, you know, as you get to the, you know, that you've differentiated a nurse cell now, you're trying to start to make a follicle, and that's when problems crop up and it just arrests. So it's very important for the differentiation of nurse cells, but it's not really, we haven't defined a function in the stem cell. But that doesn't mean that the non-canonical chromatin doesn't have some other functions, possibly involving the onset of high RNA production, you know, the onset of heterochromatin formation, or something that even happens in the stem cell that we don't know about.
Mhm. So Di Wu is asking, in mouse oogenesis, the two-wave oocytes share much similarity in transcriptome. How much are they different in growth, maturation potential, and future developmental potential? Does the small difference in transcriptome matter?
Well, it probably helps one to understand why they have such a different fate. You know, for example, we see a lot of expression of genes involved in steroid metabolism in the BPGs that are the medullar ones. It suggests to me that the hormonal milieu in the medulla may be very different than it is in the cortex, and that may be why the EPGs never want to go in there, and they halt their, you know, their movement at the cortex. Conversely, the EPGs express some interesting extracellular matrix genes, but the same one, some of the same class that are used downstream of the Drosophila stem cell to protect against the signaling from the niche, and that it could be that the primordial follicles are kind of walled off from signaling by an extracellular matrix that's produced by those cells. So it's leading to some interesting ideas. You know, I think the wave 1 follicles in the mouse do seem to produce some functional oocytes. It's thought in the humans they don't, but they may well have endocrine functions or other kinds of functions, so that it's not necessarily solely their function to produce functional oocytes. Most of the oocytes come from the stored oocytes pool of the wave 2.
So there are 2 questions to the death of the programmed cell death in mouse, one from David Cutts and one from Mary Mullins. And just to summarize them both, so Mary is suggesting that it could have something to do with the Balbiani body, and David Katz is suggesting that it has something to do with the piRNA system. So
well, I think those are both great possibilities, and it would be fun to study more, and that's, I hope we and others will do that.
So thank you, Allan. We're giving you a big virtual applause.